Okay, welcome back everyone. We're continuing on here with Chapter 6, the chapter on microbial nutrition and growth. In Part 1, we covered chemical requirements for microbial growth. Let's now look at physical requirements, starting with temperature. Temperature, as you might imagine, has a very profound effect on growth. So if we look here, we can just see the general effect of temperature on bacterial growth. So notice that the bacteria grow best right here. That's optimum temperature. That's the best. If we cool down the temperature, growth tapers off gradually. If we heat up the temperature, growth drops precipitously. And you can see here at 22 degrees Celsius, very little growth. 30 degrees, more growth. 37, which is optimum, lots of growth. Okay? Now, we can divide microorganisms into 4 basic categories based on their preferences for growth. Psychrophiles, preferring it the coldest. Mesophiles, kind of a medium temperature or body temperature. Thermophiles, liking it much hotter, and hyperthermophiles, very, very hot. Okay? It's going to be mostly archaea here, those extremophiles. So breaking that down further, psychrophiles have an optimum growth between about 5 and 12 degrees Celsius. So remember, 0 Celsius is freezing. Body temperature Celsius is about 36, 37 degrees Celsius. Boiling is 100. So they prefer that pretty cold. Some can live to minus 20 degrees Celsius. Where would you find these guys? Well, obviously very cold regions. So polar regions, very cold regions, perhaps deep sea regions. Okay? And often this red color on the ice are the psychrophiles that are growing on it. Many of these things cannot grow at room temperature. They need it much colder. They've evolved for much colder conditions. Mesophiles have an optimum growth of 25 Celsius to 40 Celsius. So this is going to be your normal microbiota. So the bacteria in and on your body, remember body temperature is about 36, 37 in here. So your normal microbiota and most everyday bacteria. Bacteria in the environment, the soil around you, those are all mesophiles. So it would also include pathogens, disease-causing microorganisms would be mesophiles, and those involved in food spoilage. Thermophiles prefer it much warmer. Optimum growth of 55 degrees to 70 degrees Celsius. That's very, very hot. Typically, they will not grow below 45. So they've evolved in much, much warmer conditions. You might find them in compost piles, that heat being, this is steam being given off here. That's the thermophiles deep in this compost pile, giving off that heat. And then hyperthermophiles have an optimum growth 85 to 95 Celsius. That's very, very warm, very close to boiling even at 100. This is going to be mostly archaea. You might find these at deep ocean hydrothermal vents or at volcanic hot springs like pictured here. So here's just a quick question. Would you be likely to find a psychrophile in your freezer? So initially we might say, oh, okay, so a psychrophile's optimum growth between 5 and 12. So yeah, my freezer's certainly cold enough. A psychrophile could live there. But would you be likely to find one there? Probably not. Why? How would it get there? Many can't grow at room temperature. So how would it get there? So it could live there, but you're not likely to find it because it's highly unlikely to get there. Okay? Now, another physical requirement is pH. Most common bacteria grow best between 6.5 and 7.5 pH. See, if you remember your pH scale, that's quite neutral. So most of your normal microbiota, most pathogens are going to grow between 6.5 and 7.5. Mold and yeast, like this picture here, molds and yeast tend to grow best between 5 and 6, so a little bit more acidic environment. You do have acidophiles, though. These are going to be microorganisms that prefer very acidic environments, optimally below 5. So Acetobacter here, this bacterial species, is an acidophile used to make vinegar, much more acidic. Helicobacter pylori is a pathogen I mentioned briefly in a previous chapter that lives in your stomach. It can lead to ulcers. Now, Helicobacter pylori is not an acidophile. So how does it survive in your stomach? Well, it lives in the lining, which is protected by the mucus that naturally protects your stomach from its own acid. Alkalophiles grow best in very basic environments. Alkaline is another word for basic. So alkalophiles grow best in pH 10, 11, 12, above that. This species here is an example. All right. Now, what about water pressure? We might remember from our basic biology that we have conditions with water pressure called isotonic, hypertonic, and hypotonic. And hypertonic environments can lead to what's called plasmolysis. Now, here's what we mean. Here's an animal cell in an isotonic environment. Isotonic means that an equal amount of water leaves, red arrow, as come in, blue arrow. So most cells prefer isotonic. That's normal. Hypotonic means more water comes in than leaves. So the cell is going to bloat, and if that continues, it'll burst. It's called lysis. You can see how this plant cell is really bulging from that. Hypertonic means more water leaves than enters, so the cell shrinks and shrivels. When we shrink and shrivel, this is called plasmolysis. We see this come in like that. So generally, cells prefer isotonic environments and work to maintain that state. What about salinity? This refers to levels of salt concentration. One group of microorganisms is called halophiles. Halophiles are organisms that live in high salt concentrations. They could be obligate halophiles, which must live in high salt, or facultative halophiles, which can live in high salt, but they're not dependent upon it. Now, a good example of an obligate halophile are those species that live in the Dead Sea or the Great Salt Lake in Utah. These organisms live in 30% salt. That's about 9 times saltier than the ocean. So this person's likely to have some obligate halophiles on her skin. Okay, now the next topic, that's physical requirements. Now, another topic that's really, really important when it comes to bacterial nutrition and growth is biofilms. We talked about these briefly before, but a biofilm is a complex community of microbes that's attached to a surface. Well, you can see it attached to a surface here and here in this pipe. Okay, I showed you a picture in an earlier chapter of a biofilm inside a catheter. All right. Now, why do microorganisms grow in these biofilms? Well, it provides them many benefits. It helps individual cells avoid environmental stresses because you can rely on other cells for protection. Cells can share nutrients. They can work cooperatively. And they can resist host defense. If they're pathogenic, they can resist defenses. So we might find microorganisms forming biofilms in the intestines, the urinary tract, sewage treatment systems, aquatic ecosystems, etc. So while you don't have to live in a biofilm, the benefits outweigh anything that might be negative. There are numerous benefits at work here. So how does a biofilm form? Well, we're going to start with a free-living microbe. So microbes that are not in a biofilm are going to then attach to a surface. When they attach to that surface, they start releasing proteins, and then the proteins mixed with the cells themselves forms this matrix, this foundation of protein and cells. Attaching to a surface to begin with is often helped by the presence of an outer layer we talked about in the chapter on prokaryotes called a slime layer. So the slime layer helps with that attachment. So I have this mixture of cells and proteins, this matrix. New cells then begin to join the community, often of different species. And eventually we reach a point where quorum sensing is involved. Now, quorum sensing refers to the point at which microbes in the biofilm reach a critical mass. They get to a point where there's enough microbes and enough different species in this biofilm where the microbes now start secreting chemicals to communicate with each other. And these chemicals can communicate things like numbers. So how big is the biofilm? How many numbers of individuals are in it? And what types of cells, what species are in there? So the microbes, in a sense, are talking to each other. They're communicating with each other. How many numbers do we have and what types of cells are here? This allows different organisms to cooperate metabolically and cooperate with each other. And it also causes the cells, once we start secreting these quorum chemicals, it causes the cells to change their shape and change their biochemistry so that they start resembling each other more and more. They become more uniform in biochemistry and shape. So here we have our free-swimming microbe that might be vulnerable to a stress, okay? They land on this surface, often facilitated by a slime layer. Now, the cells begin producing a matrix, and once they reach a critical mass, they secrete these quorum-sensing chemicals like this one. Quorum sensing triggers cells to change their biochemistry and shapes, becoming more uniform. New cells can arrive, including new species. Channels of water can flow through this biofilm. And then eventually, some microbes may leave the biofilm to perhaps establish their own elsewhere or just live on their own. Okay? So like this guy says, listen up, my fellow bacteria. We will attack together when we are enough in number. So how many are we? Start counting. And I count with those quorum chemicals. Now, what's the clinical significance of biofilms? It's estimated that 70% of bacterial infections involve biofilms. Why is that important? It's important because being in a biofilm helps bacteria defend themselves from the immune system. So it helps the bacteria shelter from the immune system. Microbes in biofilms are at least 1,000 times more resistant to antibiotics than those that are not in biofilms. So this biofilm here of Pseudomonas in the lungs is a leading cause of death in cystic fibrosis patients because they are so resistant to antibiotics. So the biofilm really helps them shelter and avoid the immune system. Okay, so now that we know about chemical and physical requirements and a little bit about biofilms, let's talk more specifically about how microorganisms can grow and how we can get them to grow in the laboratory. So what are some lab techniques for culturing bacteria? Now, most of the techniques we're going to talk about were first developed in the 1880s by this man here, a German scientist named Robert Koch. Now, what sort of media, what can we use to grow microorganisms in the laboratory? We can use an agar plate, an agar tube, a broth culture, or an agar slant, and they look like this. Here's an agar plate. Now, what is in this Petri dish, this is called a Petri dish, what's in here is nutrient agar. So this is it's semi-solid, more solid than liquid, and it's infused with water and nutrients, and also a material called agar, which is a thickening agent. Okay? So we can have an agar plate, this deep agar tube. This is just agar in a tube. We might put anaerobic bacteria down at the bottom. This is broth, which is the same thing as this nutrient agar, except with no agar in it. So it stays liquid, it doesn't harden, and you see the slant here on this agar slant is a good place to store bacteria. And it's less room than what plates take up. Now, again, as you saw in that diagram, we can use solid or liquid media. Agar is a material that we get from red algae, and it's used as a solidifying agent in culture media. It causes the media to harden and solidify. It's not like hard as a rock, but it's more solid than liquid. Agar is also used in some foods like ice creams and Jell-O as a thickening agent. Now, agar is not eaten by microbes. It's not metabolized. It's not broken down by microbes. It's not a food source. It's simply a thickening agent. Agar will liquefy at 100 degrees Celsius, and it hardens at about 40 degrees Celsius. That's when that solidifying comes in. A colony, and we could use that term for bacteria, yeast, or even molds, a colony is a physical structure that's seen on the medium. So on solid media, an agar plate or a slant or a tube, when we see the bacteria growing, that physical structure is called a colony. And the shape and appearance of a colony is often used diagnostically and taxonomically to distinguish one species from another. This is known as colony morphology. So we see bacteria growing on this agar right here. We can get these isolated colonies, and we can look at the size and shape of the colony. Now, a colony arises from a single microorganism, a single unit, called a colony-forming unit, CFU. This could be as little as one cell or perhaps a small group. And then those cells divide by binary fission, and the population grows and grows and grows. So millions of cells are in one of these dots, one of these colonies. Now remember, a colony is very different from a biofilm. Biofilms can have more than one species in them. These colonies here are just an individual, one species. You can also have pure and mixed culture. Pure culture is just one species growing versus a mixed culture where you'll have more than one. So this is a pure culture, just one species here, or mixed, and we can see the different species by colony morphology and color here. Culture media is any media designed to support the growth of microbes or cells. So any substance that supports the growth of microbes or the growth of cells. Media can be solid, it can be liquid, or it can be semi-solid, sort of jelly-like. Defined media is media in which we know all the ingredients. All the chemicals used in it are known. So there would be no yeast, no animal or plant tissue in it. Because if I just put like muscle tissue in it, I don't know exactly what's in there. So all the ingredients are known in defined media. Complex media, we know some of what's in there, but not exactly. So for example, this is a defined media. All right. Notice we know exactly the ingredients and the exact amounts. This would be a complex media. So notice one of the ingredients is just peptone, partially digested protein, beef extract. We don't know exactly what's in there, but we know approximately. Okay, we know it's beef extract. We don't know the exact chemical makeup. And then we also have what's called differential media. Differential media is used to distinguish one type of microorganism from another growing on the same medium. So it uses biochemical characteristics to make the distinction. So this is blood agar. Blood agar is an example of differential media. So we see here one species of bacteria spread on the blood agar breaks down the blood this way, that yellow color. Another breaks it down this way, more of a greenish color. And this third species doesn't break down the blood at all. So all three are growing on the same media, but we can tell them apart. That's differential media. Selective media is used to select for certain microorganisms. So some media, for example, are more selective for fungi. So some bacteria might grow on it, but mostly fungi, or vice versa. Here's another example. Let's say we want to grow a microbe that's resistant to an antibiotic. I could add that antibiotic to my media. All the other species will die, but that resistant one can grow on it. All right? So this is an example of how selective media is used. If I make the pH of this agar plate, this nutrient agar 7.3, I'm going to get mostly bacteria. Remember, bacteria prefer 6.5 to 7.5 pH. If I lower it to 5.6, I get mostly fungi. Fungi prefer 5 to 6. All right? This is mannitol salt agar, which is an example of media that's both selective and differential. Notice I have the two species of Staphylococcus growing on the same plate. This one turns yellow. This one stays red. So it's differential. I can tell them apart. This mannitol salt agar is also selective for bacteria, not fungi. Or this one, McConkey agar, an example of media that's both selective and differential. So on regular nutrient agar, my Staphylococcus and my E. coli look the same. All right? But I have McConkey agar, Staphylococcus aureus, there's no growth. Staph won't grow on it, but E. coli does. In another example, here's McConkey agar, here's my E. coli, it grows normally, and Salmonella enterica also grows on it, but it turns yellow. So selective and differential. These are carbohydrate utilization tubes as differential. No fermentation. Here's fermentation because it turns yellow, and we know it releases CO2 as a gas because I get that bubble here in this inverted Durham tube. All right? So, how do bacterial cultures actually grow, the cells themselves? Now remember, bacterial cells divide by binary fission. One cell copy its DNA, and the cell splits in half. So 1 cell to 2, 2 to 4, 4 to 8, 8 to 16, very, very quickly. So after each generation, the number of cells doubles. This is called exponential growth. And we see that here. Boom, 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 boom. So we're doubling every generation. All right? So you don't get a straight line. If we double every generation, we're going to get this J-shaped curve where we're doubling every generation. So, if we plot the growth of a bacterial culture over time, you can create what's called a bacterial growth curve. Exponential growth, that J-shaped curve, eventually levels off. And when that J-shaped curve levels off up here at the top, how bacteria actually grow because of that leveling off is called logistic growth, or sometimes logarithmic, but we'll say logistic growth. There are 4 parts to the bacterial growth curve: a lag phase, a log phase, a stationary phase, and a depth phase. Now you should know this curve. All right, so here's time down here. We're going through time, and this is the number of cells in my colony. Now the lag phase is the beginning. That's slow at first, so only 1 cell to 2, 2 to 4, 4 to 8. You can hardly detect any change. But as we keep doubling, eventually we're going to see that exponential growth, this J part of the curve. That's the log phase. Now, eventually this levels off because resources get scarce. When this levels off, this is called the stationary phase. And eventually, as resources continue to get scarce and waste products build up in my now large colony, death starts to occur and my population drops. So that's my death phase. Generation time is the amount of time it takes for the number of cells to double, or it's sometimes called doubling time. We would measure that here in the log phase when we have active exponential growth. So here's a problem. If you put one bacterial cell in nutrient broth and that organism has a generation time of 20 minutes, so every 20 minutes the population doubles, how many bacterial cells would be in the broth after 5 hours? Now, the first thing we're going to want to do is calculate the total number of generations. So 5 hours total, right? But it's 20 minutes for a generation time. So let's make everything in minutes. So 5 hours equals 300 minutes, right? 60 times 5, 60 minutes in an hour. So 300 minutes. So now I'm going to take 300 divided by my 20-minute generation time. So in that 5 hours, there were 15 generations. So 15 times my population is going to double, all right? So since I'm doubling the number every generation, all I have to do is raise 2, the number 2, to the power of generations. So I'm going to get out my calculator and I'm going to put 2 to the 15. Okay, 2 times 2 times 2 times 2 times 2 times 2, all the way up to 15 times. And if you put that in your calculator, that's 32,768 cells. So you should know how to calculate this generation time. During which growth phase would I want to measure this? Again, that's during the log phase. The bacterial cells are actively dividing. Now, real quick here, what about fungi? Yeast are grown in the lab using similar methods to what we just talked about. Like bacteria, yeast grow in colonies on agar plates. Their colonies tend to be a little bit bigger, okay, because this is a eukaryote. They have distinct colony morphologies, different species of yeast. We can look at the morphology, the color and shape. They reproduce asexually by a process called budding. One cell, a parent cell, buds off a small new cell. So they can grow rapidly, not quite as rapidly as the bacteria. Mold can grow on agar plates, either by hyphae, remember the threads, the chains of living threads. So if we break off hyphae, put it on an agar plate, the hyphae can start to grow. This is asexual. Or you can have spores. The mold may form spores, small packet of DNA surrounded by a tough outer layer. Spores can be formed asexually or sexually. So mold spores forming mold spores forming from penicillin here. This is spores forming from another species here, and the hyphae, and 4 different species of mold growing on the same agar plate. Now, fungi tend to reproduce sexually if there's bad conditions, unfavorable, but switch to asexual if conditions are more ideal. Now, this is successful because if you have bad conditions, you're going to want diversity. Sexual reproduction provides diversity. So in bad conditions, if there's diversity, some individuals might survive better than others, and we don't go extinct. If you have really, really good conditions, we're going to want to switch to asexual because it's easier and faster. Okay? All right, everyone, that ends part 2 of the chapter on microbial growth and nutrition. Next time, we're going to look at a little bit deeper into this. We're going to talk about microbial metabolism, probably do that in 2 parts as well. So that is it for now. Until next time, have a nice rest of the day, and we'll see each other again soon. Bye-bye.